Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the Histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below.
Hello and welcome to the Respiratory System Histology. In the first of the two lecture series, we'll look at the general anatomy and histology of the respiratory system and look at the detailed histology of the upper respiratory tract. Anatomically speaking, the respiratory system is comprised of a series of tubes that carry the air from outside down to the lungs. And within the lungs, we have these numerous and small spherical structures called alveoli, where the gas exchange can actually occur. The alveoli have really thin walls that have tiny little capillaries on the outside. So the gas exchange is occurring between the air and blood across this thin membrane. So from that perspective, the respiratory system can be divided into the conducting portion and the respiratory portion anatomically. The conducting portion is the structures that carry the air from outside down to the gas exchange surfaces. So we're talking about the nares, the openings at the bottom of our noses, and the nasal cavity, pharynx, larynx, the trachea, the bronchi, and all of their branches down to the smaller or smallest of their terminal branches called the terminal bronchioles. And the respiratory portion then is the structures where the gas exchange can occur. So we're talking about the things like the respiratory bronchioles, which are even smaller than the terminal bronchioles, and its walls are actually interrupted by the alveoli. And in between the respiratory bronchioles to alveoli, there are some alveolar ducts and alveolar sacs that lead into lots and lots of alveoli, which we'll look at in the later sections. Now, while we're on the subject of anatomy, it is also important to note that outside of all these tubes and alveoli, we also have this important structure called the diaphragm. This is this dome-shaped or umbrella-shaped, thin, muscular tendinous sheet. Its bottom parts are all anchored to the rib cage and to the back body wall. When the diaphragm contracts, this dome shape actually comes down a bit like this, thereby increasing the thoracic capacity. It inflates the lungs, so that's our inhale. When the diaphragm relaxes, the dome goes back up and reduces the thoracic volume, therefore squeezing our lungs, and now we are exhaling. This process is involuntary because we don't have to think about every single breath we take. There are some other muscles kind of in and around our body wall that we can consciously contract and relax a little bit more to kind of increase or decrease that thoracic volume a little bit as well. And another very important aspect about the respiratory system is the fact that it works so closely with our cardiovascular system. So there's the heart right here, which I'll kind of draw in a non-anatomically correct position and shape. But we all know that the right side of the heart receives all the deoxygenated blood from the body, and all that deoxygenated blood is then pumped out by the right ventricle into the pulmonary trunk, then into the pulmonary arteries, which then go into the right and left lungs. This deoxygenated blood are then carried by smaller and smaller arteries right here, and then they get down to the capillary networks that are outside the alveoli. And that's where the gas exchange occurs and our blood gets reoxygenated. Then all that reoxygenated blood will then drain into the pulmonary veins, which then are returned to the heart into the left atrium. And off of the left atrium, we have aorta, which carry all that oxygenated blood down to the rest of the body, into the systemic circuit. From that perspective, we do have a little bit of a problem in that the majority of the blood coming into the lungs are actually deoxygenated. So in order to support the tissues of the lungs themselves, we need secondary blood supply coming from the systemic circuit. So from that sense, our lungs have dual blood supply. One of the two blood supplies coming into the lungs again is coming from the pulmonary trunk off of the right ventricle ventricle, which then divides into the right and left pulmonary arteries, carrying deoxygenated blood into the lungs. Then off of the aorta, which travels behind the heart along the back body wall like this, will send out little branches of arteries called the bronchial arteries into the lungs. And these are the arteries that deliver the oxygenated blood to the lung tissues. Looking at the histology of the respiratory system, in kind of general terms, we know that the respiratory tract is really nothing more than a series of tubes with the wall composition that can vary depending on their location and function. So for example, in the nasal cavity, we'd be a lot more concerned about carrying a lot of air or sucking down a lot of air from the environment and perhaps filtering it of any microbes, big dust particles, and any particulate matter, as well as humidifying and warming or cooling that air depending on what kind of environment we're in. The distal part would be more concerned about now gas exchange of already conditioned air that's come down. But despite some of these variations throughout the respiratory tract, there are some features that are shared throughout the respiratory tract. One is the mucosa. Mucosa is a general term that refers to any kind of inside lining of our body. So anywhere inside the body that is exposed to the external environment, like the respiratory tract, our gastrointestinal tract, and urogenital tract. Those are all lined by the mucosa. And I call this the skin inside the body. And much like how our skin is comprised of two layers of tissues, the lining epithelium and the underlying connective tissue, mucosa inside the body is also comprised of the lining epithelium and the underlying connective tissue, generally termed the lamina propria. So think already about what kind of epithelium would line our nasal mucosa, perhaps the pharynx, larynx, and the trachea, down to the alveoli. The lamina propria, generally speaking, is comprised of loose connective tissue in the respiratory tract. It would be rich with neurovasculature, but also it would contain some mechanisms for monitoring the external environment and for responding to any potential microbes or pathogens that the mucosa may encounter. So expect to see a lot of immune cells spread throughout lamina propria, as well as potentially some aggregates of these immune cells. Outside of the mucosa in the respiratory tract, there are some additional supportive structures, whether it be the bones, cartilages, smooth muscles, or even extracellular fibers. They're there to provide architectural support of this tubular structure in order to keep the lumen open usually, and to regulate how much air can come in or outside of certain regions. And depending on which part of the respiratory tract we're talking about, there are some accessory structures present to provide some additional and specific support. There could be some glands in the lamina propria or even deeper regions of that respiratory tract wall to produce seromucous secretions in order to keep the lumen nicely humidified and wet, and even to propel some of the foreign particles that have been inhaled back up into the upper respiratory tract. There are also lymphoid nodules, as mentioned earlier, the aggregates of immune cells in the lamina propria or even in the deeper layer of the respiratory tract wall. These type of lymphoid nodules are not unique to the respiratory tract. They're also found in the gastrointestinal tract and urogenital tract as well, that are exposed to the external environment. We call the lymphoid nodules that are associated with any kind of mucosa throughout our body the mucosa-associated lymphoid tissue, or MALT in short. So expect to see lots of MALT, not only throughout the respiratory tract, but in other systems as well. Alright, now let's take a histological tour of the respiratory tract, beginning with the conducting portion. Of course, we all begin at the upper respiratory tract with the external nares and the nasal vestibule. The external nares are basically our nose holes where the air enters the nasal vestibule. The nasal vestibule is this funnel-shaped area immediately inside of the external nares. So we're expecting to see the skin turning into the mucosal histology histology here. So from the outside, we'll see the keratinized, stratified, squamous epithelium of the skin. Now in the inside, in the nasal vestibule, that keratinization would go away and we're seeing the non-keratinized, stratified, squamous epithelium. Within the nasal vestibule, there are a lot of coarse hair that extend out into the space, and these are called the vibrissae. The vibrissae are covered by a lot of serum mucus secretions that are in the lamina propria of the nasal vestibule. And these serum mucus secretions would cover the vibrissae, and together the hair and the glandular secretion would be very effective at capturing a lot of kind of larger dust particles or particulate matters in the air before they even enter our nasal cavity. The nasal cavity then is even posterior to the nasal vestibule. And this enlarged space is actually compartmentalized by first the nasal septum. This is a bony ridge or wall that divides the nasal cavity into right and left nasal cavities. While the nasal septum has a bony core in the middle, it is covered on the exterior by the mucosal lining, of course. And when we remove that nasal septum, there are these shelf-like structures on the lateral walls of the nasal cavities on both right and left side that are called the nasal conchae. And these lateral shelvings or extensions come out towards the midline of the nasal cavity. And again, the nasal conchae have bony core for structural support, but on the exterior, they're all covered by the mucosa. The nasal conchae as a whole serve an important role in creating turbulence in the incoming air. It also increases the surface area for contact between the mucosa and the air, for that air to have an opportunity to humidify and warm or cool depending on the external environment. So when we look at the histology of the nasal cavity, we'll see that the mucosa is lined by the respiratory epithelium up here, which is the ciliated pseudostratified columnar epithelium. Deep to the respiratory epithelium, we have this layer of lamina propria, the connective tissue, and even deep to this layer called submucosa, there are these lobulated and lobar organization of serum mucus glands, which release their products into these ducts, then into the nasal cavity. So these glands play an important role in humidifying the incoming air. Furthermore, there's a lot of vasculature in the lamina propria as well as in the submucosa that get dilated so that the incoming air have the opportunity to be warmed or cooled by the constant temperature of the blood that's in the lamina propria. Before we move on, we need to make a point about the superior nasal conchae. The right and left superior nasal conchae are actually covered by a specialized sensory structure called the olfactory epithelium, as opposed to the respiratory epithelium, which means the olfactory epithelium contains a lot of specialized sensory cells that can detect the olfactants that are coming into the nasal cavity. And lo and behold, there's this big fat olfactory bulb of the cranial nerve number 1 sitting right above the superior nasal concha with the neural input going in and out of that superior nasal conchae. So when we look at the histology of the superior nasal concha, it'll look something like this. We're kind of at a macroscopic view here, but what we see is this elaborate curvy structure that is made up of the bony core in the middle and covered on the exterior by the mucosa on either side. What you may notice from this macroscopic view is that the lining epithelium on this side and even on this side and this side, these are all pretty thick compared to the lining epithelium on this side. And it's because we're actually seeing the respiratory epithelium here that is transitioning into the olfactory epithelium right around this area. So superior nasal concha has the olfactory epithelium that is continuous with the respiratory epithelium. Looking at the boxed area at a higher magnification, where we can actually see the respiratory epithelium right here, which is made up of thinner layer of pseudostratified ciliated columnar epithelium, which is starting to transition right around here. That same epithelium is starting starting to thicken. And on the other side, right over here at higher magnification, we can see that that olfactory epithelium is made up of still the ciliated, pseudostratified, columnar epithelium, except that this epithelium is much thicker than that of the respiratory epithelium or their counterpart down here. That has to do with a lot of tall special sensory cells that's occupying this olfactory epithelium. Another interesting thing to note about the olfactory epithelium and the neighboring respiratory epithelium is the composition of the lamina propria. In the respiratory epithelium, the lamina propria, it's vascular, it contains a lot of connective tissue fibers and things like that, but it is fairly small or thin. Compare that to the lamina propria of the olfactory epithelium above. It contains a lot of these glandular structures that are kind of looking bulbous. These structures are called the Bowman's glands. They're specialized for producing a lot of watery secretions that'll not only dissolve the olfactants that are floating around in the nasal cavity, but also to wash those olfactants away so that the special sensory cells are always kind of refreshed and renewed and ready for further olfactants to come in and bind them. Some other things we see in the lamina propria of the olfactory epithelium include these neural structures that carry those special sensory fibers back to the olfactory nerve, and there are of course lots and lots of vasculature within the lamina propria as well. So once again, we're seeing the gross anatomy at the histological level, and now we're able to incorporate the functional perspective to this whole macroscopic structure as well. Let's continue our journey down the conducting portion of the respiratory tract, which brings us to the pharynx. Now, this is this large space on the posterior aspect of our head and neck area, and this area is subdivided loosely into three general regions. The nasopharynx, which is a space that is just behind the nasal cavity, the oropharynx, which is this area just behind the oral cavity, and laryngopharynx, which is the space that is below the oropharynx. And again, all the pharyngeal space is lined by the mucosa, but depending on which area of the pharynx we're talking about, the lining epithelium will be slightly different, because what travels through these three different spaces can be quite varied. For example, nasopharynx would be lined by the respiratory epithelium, because for the most part, the only thing that'll travel through the nasopharynx would be the humidified air, as opposed to the oropharynx, where we're not only conducting air from the nasal cavity, but also some food bolus from the oral cavity as well. So to accommodate different contents of the food or fluid bolus coming from the oral cavity, the oropharynx is actually lined by the non-keratinized, stratified, squamous epithelium. The laryngopharynx then is potentially a common space also for air as well as food bolus. So for the most part, this area would be lined by the non-keratinized, stratified, squamous epithelium as well. Moving down the conducting portion now into the larynx, which is the last part of the upper respiratory tract. Larynx is this complexly shaped, cartilaginous, circular structure through which the air is directed into the lower respiratory tract. So larynx can function as a gateway into the lower respiratory tract with this specialized structure called the epiglottis, which when it functions properly, it stops food or fluid boluses from going into the lower respiratory tract. Furthermore, there are structures inside the larynx called the vocal cord, which I am vibrating right now to generate voice. So with these different types of functions, the larynx has quite the complex anatomy. So let's spend a few minutes looking at the anatomy of the larynx. Larynx is comprised of a number of cartilages that provide structural support to this complexly shaped anatomy. Two of the most prominent cartilages are the thyroid cartilage and the cricoid cartilage. The thyroid cartilage is this big shield-like structure on the anterior aspect. This structure doesn't go all the way around to form a ring. And it is the thyroid cartilage that has this kind of anterior and superior prominence that most people call the Adam's apple. Anatomic name for this structure is the thyroid prominence. And this is where the vocal cords attach. So the longer vocal cords you have, the deeper the voice will get. Hence the reason why the thyroid prominence gets larger or longer or more prominent in males at puberty than in females. The growth of this structure is under male sex hormones. Thereby the lengthening vocal cords can generate some interesting pitches and sounds of voice for a time being until that growth is complete, and now we're left with much deeper voice than one may have started out with pre-puberty. The cricoid cartilage is located inferior to the thyroid cartilage, and this structure is actually a complete ring, but the anterior aspect is a little bit smaller or narrower. But as this cartilage goes towards the back, it actually becomes elaborated or more broadened to form a complete ring with proper backside that can then keep the larynx open. Looking at the two cartilages from kind of anterior / just a little bit oblique perspective, here's the thyroid cartilage looking like a shield anteriorly, and here's the thyroid prominence, and underneath or inferiorly we have the cricoid cartilage that'll go all the way to the back. And from the backside of that larynx, so we're looking at the posterior view here, the thyroid cartilage kind of ends like right around here, while the cricoid cartilage goes all the way to the back and becomes a big structural support for that laryngeal opening. When we make a mid-sagittal section right through this larynx and look at the cut plane, here's the anterior aspect and here's the posterior aspect, we're able to see the cut plane of the thyroid cartilage here. That doesn't go all the way to the back, but the cricoid cartilage, we can see how this cartilage forms a complete ring and becomes a much larger, broader structural support in the back. Another cartilage that plays an important role in the larynx is the epiglottis. And this structure is shown right here in this image, as well as from the posterior view, we can see this leaf-like or even petal-like structure that is the epiglottis. From the sagittal view, the epiglottis is this flap-like structure that expands up from the anterior aspect with its expansions going laterally like that. And what this epiglottis does is quite important. Every time we swallow, this epiglottis will bend downward like this and cover the opening of the airway. So all the food and fluid bolus that we swallow will go down to the esophagus that is positioned posterior to the lower respiratory tract. And when we're not eating or swallowing, then the epiglottis will flip right back up into its original shape so that all that air can get into the lungs. There are also 3 additional pairs of cartilages that are on the backside of the larynx that play a role in different pitch generation when we talk. Other very important structures of the larynx are the true and false vocal folds, also known as the vocal cords. These folds are these protrusions of ridges of tissues that are extending into the lumen of the larynx. We of course have the right and left side, so these are paired structures. The superior protruding tissue is called the false vocal folds, whereas the inferior protrusion of tissues is called the true vocal folds. So it's the true vocal folds that are constantly vibrating in order to generate sound. The false vocal folds contain a lot of seromucous glands whose products are able to coat the inside of the larynx and keep this environment nice and moist and protect the true vocal folds. Now then, having learned the anatomy and some functions of these different parts of the larynx, let's predict the types of cartilages that we might find in the thyroid cartilage, the cricoid, as well as the epiglottis. Since the thyroid and cricoid cartilages have more of a structural role in keeping the larynx open, these cartilages are going to be made up of the hyaline cartilage, which is rigid. The epiglottis, however, needs to bend and recoil back to its original shape every time we swallow, so epiglottis would be made up of the elastic cartilage. And of course, all of these cartilages aren't just hanging out and exposed to the air. They're all covered, especially in the areas that are exposed to the external environment, by the mucosa. And speaking of the mucosa, let's predict the types of lining epithelia in these different areas of the larynx. What type of epithelium do you expect to see covering the superior aspect of the epiglottis versus the inferior aspect of the epiglottis? What about the lining epithelium of the false versus the true vocal folds, given their functions? Let's look at these histology in a little more detail in the next few slides. Let's first look at the epiglottis in a little more detail. Here's the histology of the epiglottis. From the macroscopic view, we're able to see these plates of cartilage that's forming the core of this structure. So all of these structural core of the epiglottis would be made up of the elastic cartilage that is of course covered on the outside by the perichondrium. Now what about the lining epithelium of the epiglottis? Let's look at the tip of the epiglottis, and this is what we would typically see. Here's once again definitely the elastic cartilage forming the core of the epiglottis, and the perichondrium on the outside, some lamina propria or loose connective tissue here, and covering this entire structure on the outside would be the lining epithelium. What tissue do you think that is? And even without zooming in any further, I hope you're seeing the non-keratinized stratified squamous epithelium. And that should make sense because this structure comes in contact with both the bolus from the oral cavity as well as the air coming from the nasal cavity. So this area needs to withstand a lot of force and friction. So the portion of the epiglottis, including the tip, and the portion that we call the laryngeal surface, that is facing the oral cavity and laryngopharynx, are all lined by the non-keratinized, stratified, squamous epithelium for strength and protection. Now what about the underside of the epiglottis, the side that doesn't necessarily come in contact with the bolus from the oral cavity? This area is most commonly lined by the respiratory epithelium, the ciliated, pseudostratified, columnar epithelium. Now what about the true and false vocal folds? In this sagittal view of the larynx, we reviewed that the lingual surface as well as the tip of the epiglottis is lined by the non-keratinized, stratified, squamous epithelium, but the underside, the laryngeal surface of the epiglottis, is lined by the respiratory epithelium. So then what about the rest of the larynx? There's true and false vocal folds. What type of lining epithelium would we expect to see? Now here's histological section through the larynx at this plane. So this would be the luminal side, this would be superior aspect and inferior aspect of the larynx. This protrusion is the false vocal fold, and this is the true vocal fold. Looking at the false vocal folds at a higher magnification, this is the histology we're seeing. Here's the lining epithelium and the underlying lamina propria, and even deeper to that, we have the submucosa containing lots and lots of serum mucus glands in lobular and lobar conformation. So we can see that the false vocal folds contain lots of glands that can produce the fluid that'll coat the inside lumen of the larynx. The lining epithelium-wise, the false vocal folds are covered with the respiratory epithelium consistent with majority of the lining epithelium of the larynx. The true vocal folds, however, when we think about it, we're constantly vibrating this region and lots of air, and as the air passes through, that would generate lots of friction and force applied to this area. So even before we start to zoom in, we can see that the lining epithelium is transitioning a bit to the point where the tip of that true vocal fold appears to be much thicker, and then it transitions back down here into a much thinner lining epithelium. So we can expect to see the type of epithelial tissue that might be good at withstanding a lot of force and friction, and here's the magnified version of this area. And lo and behold, lining epithelium is the non-keratinized, stratified, squamous epithelium. Here's the higher magnification view. Another thing that we can appreciate here is the fact that there's no seromucous glands associated with the true vocal folds. Instead, there's a lot of ligamentous connective tissue down here, once again to reinforce the true vocal folds with some strength. And a little deeper within the true vocal folds, if we were to look at this area at a higher magnification, the tissue that we'll see is the skeletal muscle tissue, which again makes sense. These are the vocalis muscles that are innervated by the recurrent laryngeal nerve, so we can consciously modulate the level of contraction of the vocal folds so that we can actually, to a certain degree, modify our voice a little bit higher pitched or lower pitched as we please.
Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.